Hello and welcome to my explanation of meiosis and the steps in the process of meiosis. Meiosis is a reproductive process that the human species and many other species can continue to exist. It is a sexual reproduction process and starts with an egg and sperm, which is why you will later see sets of chromosomes sharing genetic information in the form of polka dots of the other colored chromosomes. One pair belongs to the mother and one the father. Some info you'll want to know before we begin is what a homologous chromosome is. A homologous chromosome is two chromosomes that match in length and a centromere position. Usually they pair up, one pair from the father and one from the mother. This pair of homologous chromosomes makes a tetrad. Our first step is prophase. Keep in mind there is a prophase 1 step and a prophase 2 step. This is prophase 1. In the prophase 1 step, the chromosomes condense and the nuclear envelope breaks down. During prophase, the tetrads will swap genetic information. This is also known as genetic recombination or crossing over. Now on to the second step, in meiosis, metaphase 1. In metaphase 1, pairs of homologous chromosomes, or tetrads, will move to the equator of the cell. Once there, spindle fibers from the centrioles will latch onto the homologous chromosome and begin to separate them. These tetrads will then become diploids. A diploid is when the cell contains two complete sets of chromosomes, one from each parent. In anaphase 1, homologous chromosomes are pulled by the centrioles to the opposite sides of the cell. Now on to telophase 1 and cytokinesis. During telophase 1, chromosomes gather at the poles of the cells. At this point, the nuclei are beginning to form around the chromosomes. During cytokinesis, the cytoplasm divides and the cells are beginning to fully divide and separate, making two newly formed cells. At the end of telophase 1 and cytokinesis, we should end up with two newly formed cells and the process can begin again. During prophase 2, the nuclear envelope of both cells break down, and the centrioles begin making spindle fibers to attach to the chromosomes in the cells. During metaphase 2, chromosomes line up at the equator once again, and the spindle fibers latch onto them. Now on to anaphase 2. In anaphase 2, the centromeres divide, and the chromosomes are then separated and become haploids. Haploids are what happens when the chromosomes are no longer in pairs and are now one singular chromatid. Similar to telophase 1, and the first time cytokinesis happened, the chromosomes gather at the poles of the cells, and at this point the nuclei are beginning to form around the chromosomes. During cytokinesis, the cytoplasm divides and the cells are beginning to fully divide and separate. Now, instead of making two newly formed cells, we officially have four. As previously stated, rather than having two newly formed cells, at this point we should have four newly formed cells. We have now reached the end of my drawn model of meiosis. My supplies included whiteboard, dry erase markers, and many Tupperware bowls as stencils for my hopefully perfect looking circles. Thank you for watching and listening.